I'm going to talk to you about the effect of temperature on Cots and why considering climate change is super important for management. So, two of the greatest threats to the Great Barrier Reef are climate change, resulting in mass coral bleaching events, and crown-of-thorns starfish. So, as Roger said yesterday and Dan said today, these two threats can have cumulative or synergistic effects on the reef. However, effective management may at least reduce the threat of crown-of-thorns starfish. However, effective management would benefit from understanding how climate change is impacting Cots now in the form of the increased prevalence and intensity of heat waves, and also how sustained warming will impact Cots going into the future. So laboratory-based studies to date have found that 2 to 4 degrees of warming, or temperatures of around 30 to 32 degrees, have resulted in reduced egg size, fertilization success, proportion of embryos and larvae undergoing normal development, larval survival, and gonadosomatic index. So this diagram is from a paper by Pamela Camia, and it shows that Cots larvae in a 30-degree treatment are significantly smaller at day 4 compared to a 26 and 28-degree treatment, and at day 10, no larvae at all survive in this treatment. However, other studies have found increased larval survival, larval development rate, and juvenile growth at the same degree of warming. However, what's not so clear is how warming affects adult Cots, and this is important to know considering that this is the stage which causes the damage to the reef. And this is the stage that I'm going to focus on here. So a good way to look at thermal sensitivity is by visualizing physiological thermal performance curves. So it's generally considered that physiological performance increases up until a certain temperature, and this is followed by a sharp decline. In terms of metabolic rate, for instance, at a mechanistic level, the faster rate of chemical reactions, there is a faster rate of chemical reactions at high temperatures, which means that more ATP is required to fuel cellular processes, resulting in a greater demand for oxygen. However, at a certain temperature, organisms are unable to take in enough oxygen to meet this heightened metabolic demand, resulting in a switch to anaerobic metabolism, and this is what causes this decline we see here. And after extended exposure, there are additional things that happen, like protein denaturation, oxidative damage, mitochondrial failure, which further result in performance declines. However, the shape of this curve may vary depending on a number of factors, such as resource availability, for instance. So my first aim was to assess the metabolic responses of adult Cots to acute warming at both the organism and biochemical levels. So this is an important first step to understanding the thermal sensitivity of Cots and gives us a good mechanistic understanding behind physiological performance. So the Cots for these experiments were collected from the central mid-shelf of the Great Barrier Reef at the start of 2020. So to look at organism level responses, I used intermittent flow respirometry to establish oxygen consumption rate, which is a proxy for metabolic rate. And for this, I ramped temperatures up from 24 degrees to 36 degrees over the course of a day. So intermittent flow respirometry generally consists of a measurement period where chambers are sealed and oxygen depletion is measured over a particular amount of time. And this is followed by a flush period to remove oxygen depleted water and replace it with fresh water for the next measurement period. So in these experiments, the measurement period was 30 minutes, and this was followed by a 30-minute flush, at which point the temperature was increased to the next increment for the next measurement cycle. And this equated to about a degree of warming per hour. And as you can see, this is a picture of the setup of the experiment here. And to assess biochemical level responses, I used spectrophotometry to measure the maximal enzyme activity of both citrate synthase, which is a proxy for aerobic metabolism, and lactate dehydrogenase, which is a proxy for anaerobic metabolism. And it's worth noting that the tissue samples used for this experiment were collected in situ during the same trip as the previous set of experiments, but were from a separate cohort of starfish. And the temperature ranges used for this were 15 to 40 degrees for citrate synthase and 20 to 45 for lactate dehydrogenase. So here are my results for my organism level responses. So as you can see, I separated into two size groups, and this was because two clear size groups could be distinguished, so below 300 grams and above 700 grams, and clearly there was a strong size effect here. As you can see in small individuals, oxygen consumption rate increased throughout the whole temperature range. There was some indication of metabolic depression after 35 degrees, however. Although in large starfish, there was clear metabolic depression after around 33 degrees, indicating that larger starfish may be more sensitive to thermal extremes. So in terms of biochemical level responses, the activity of citrate synthase, which was the proxy for aerobic metabolism, declines throughout the temperature range and was negligible at around the temperature that metabolic depression was observed at the organism level. However, lactate dehydrogenase increased throughout the whole temperature range, and this indicates an increased reliance on anaerobic metabolism at higher temperatures. And this is unsustainable considering that anaerobic metabolism is about 18 times less energy efficient than aerobic metabolism, meaning that it won't be able to be sustained for a long period of time. So if aerobic metabolism can't be reinstated, this will likely result in the death of the organism. It's also worth noting that these studies were carried out on the acute scale, and there are likely additional costs associated with longer-term exposure to temperature, meaning that thermotolerance may be lower if exposed to a longer period, if exposed to temperature for a longer period of time. So this brings me to my next aim, which was to assess the effect of prolonged warming on the performance of Cots. So for this, I had 4 temperature treatments: a 26-degree controlled treatment, a 28 and 30-degree treatment, which represents the average and the maximum summertime temperatures in the collection vicinity, and a 32-degree treatment, which is a temperature predicted to be more common on the reef under near-future climate change. So these Cots were collected during the same trip as the ones collected during the acute experiments, and the starfish were acclimated at 26 degrees for 3 weeks prior to the temperatures being ramped up to the correct endpoint temperatures at a rate of no more than 0.5 degrees per day. So after endpoint temperatures were reached, at various time points, oxygen consumption rate was again measured using intermittent flow respirometry again, where oxygen depletion was measured over 30 minutes. And also survival was analysed. So from day 1 until around day 58, when the starfish were removed from the treatment, any mortality was noted, and this was later plugged into survival analysis. It's also worth noting that these starfish were starved throughout the entirety of the experiment, and of course, results may differ to some extent if the Cots were provided with food. So here are my results for oxygen consumption rate. So I found that oxygen consumption rate was 115% higher in the 30-degree treatment in orange compared to the 26-degree treatment, which is in blue. However, it was 22% lower in the 32-degree treatment in red compared to the 30-degree treatment, indicating there was some evidence of metabolic depression. And as you can see, the pattern here is similar to the thermal performance curve I showed you earlier, with an increase followed by a decrease in metabolic rate. It's also worth noting that metabolic depression was observed at around 32 degrees compared to, on average, around 34 degrees in the acute studies, meaning that time may be an important factor in thermal tolerance. And here are my results for survival. This is a Kaplan-Meier survival plot, and as you can see in the 26-degree treatment in blue, there's no death, while the 32-degree treatment in red has a lot of death, and death started occurring pretty quickly, within days of the 32 degrees being reached, meaning that it doesn't really have to be at this temperature for very long for mortality to result. And even in the 30-degree treatment, about a quarter of the starfish died. So in conclusion, temperatures of around 32 degrees result in increased mortality, which may be in part explained by the observed metabolic depression. And this is a temperature that's already been experienced on the reef in certain areas during extreme heat waves, and is expected to be more common on the reef under near future climate change. And a reduced performance in a warmer ocean may lead to population contractions or potentially redistributions to more southern cooler reefs, facilitated by larval dispersal over successive generations. It is worth noting that these studies were carried out in a controlled setting, and there are likely additional things going on in the field that will affect thermal tolerance, such as food availability, and whether the starfish may have the ability to acclimate to short-term marine heat waves or adapt over generations to prolonged warming. And this brings me to my future work and management implications. So a question we hope to answer is, how fast can Cots adapt to warming? And a first step to understanding this is conducting similar experiments to what I've already done, but comparing Cots from different populations, from different latitudes that have obviously been in different thermal environments to see whether thermal tolerance varies. So if Cots can adapt to warming, they could persist for years to come, meaning that efficient and effective management is vital. Another question is, will Cots eat more coral at higher temperatures? It's possible that due to the greater metabolic demands, as I showed you before, they will eat more coral to compensate for this. And this could also mean that Cots will be able to persist and could be an issue for the preservation of the coral that will already be threatened by climate change directly. And finally, will Cots be harder to cull at higher temperatures? It's possible that they may be more cryptic at higher temperatures, meaning that they're harder to find. As Morgan mentioned earlier, they're pretty hard to find as it is. And it's possible that they may hide during heat waves and then emerge in the winter and go on a killing spree. So this is important, obviously, to know for surveillance and control. So I would like to thank these people and also funding from the ARC Centre of Excellence for Coral Reef Studies for funding my work to date. Thank you very much.